This malaria, I am Dr. Mutha, Department of Microbiology, SMPT, IMS and RC. So the name malaria is mal means bad and area means air. So this is spread by air pollution through stagnant water and marshy lands. So the Alphonse Lavrin discovered causative agent of Plasmodium RBC of a patient in Algeria in 1888, and Sir Ronald Ross in 1897 discovered sexual cycle of parasite in mosquito in Secunderabad in India, and both got Nobel Prize for the contribution to malaria. The causative agents of malaria, previously there were four, now it is five. So first is P. vivax, it causes benign tertian malaria. So benign tertian means it is benign, so not very complicated, and tertian means the periodicity of fever is every 48 hours, that is every third day. P. falciparum malignant tertian, so periodicity of the fever is every third day. Then, but it is with the complications. Then P. malariae, so benign cotton malaria, so fever spike is every fourth day, that is 72 hours. P. oval, it is similar to vivax, that is benign tertian malaria, and the fifth species new one is added, that is P. nonaceae, so it is quotidian or simian malaria, so every alternate day fever, that is once in a 24 hours. So it is parasite of monkey but can infect humans also. So it is considered as a fifth agent. Now, transmission and life cycle of malaria. So definitely host is female Anopheles mosquito because sexual cycle, that is sporogony, takes place in the mosquito. And intermediate host is man because asexual cycle, that is schizogony, takes place in the man. Infective form, the sporozoites. Those sporozoites enter man after the bite of mosquito and the life cycle continues. Portal of entry is bite through the skin. Mode of transmission, bite of the infected mosquito, and site is first in the liver cells and then in the RBCs. The life cycle, first mode of transmission is bite of the female Anopheles mosquito, you already saw, and rarely it can be through the blood in blood transfusion or transplacental forms. In that is infective form is tropozoites or merozoites. So this picture is showing you life cycle of malaria. So here two phases are there, one asexual phase in man and then sexual phase in mosquito. So in man there are mainly two stages, that is pre-erythrocytic schizogeny and erythrocytic schizogony, and in vivax and oval you can see exoerythrocytic schizogony, and in mosquito after that it is gametogony, and then in mosquito it is sporogony. So we will see it in detail. So in this picture, we will see the two cycles in man and mosquito in detail. So first, after the bite, sporozoite enter the skin, and then through the blood circulation, they go to liver cells. There they develop into the schizont, that is merozoites, and then merozoites develop into the tropozoite and schizont. So this is called pre-erythrocytic schizogony or pre-erythrocytic schizont formation. Then some merozoites in vivax and oval, they go in dormant stage, they are called as hypnozoites, and after some days or months or years, again they may cause infection, that is called as relapse. Then the merozoites, after schizont ruptures, the merozoites are released and then they infect RBCs. Again, merozoites will develop into tropozoite, schizont, and it is called erythrocytic schizont containing merozoites. So again, merozoite will rupture and then merozoites, the schizont will rupture and merozoites are released. So these merozoites will again reinfect RBCs, or some of them may convert to the sexual phase, that is gametocytes. So male gametocytes and female gametocytes. So male gametocyte is called as microgametocyte and female is called as macrogametocyte. So at this stage again the mosquito will bite and these gametocytes, male and female, they will enter into the mosquito. Now cycle in mosquito, it is called as sporogony. So these the micro and macro gametocytes, they will develop into macrogamete and microgamete. So microgamete is having around 8 flagella. So it undergoes X flagellation and each one forms the microgamete, and then it will fertilize with macrogamete, and then it will form zygote. Then from zygote it will form ookinete, Then ookinete, it will develop into oocyst in the midgut of the mosquito, and then oocyst containing sporozoites will reach to salivary glands. Then oocyst will rupture, and then all the sporozoites are liberated in the salivary gland, and with the bite to the humans, this sporozoid form, that is infective form, is entering the human skin. So this is all about cycle of plasmodium in human, that is schizogony, gametogony, and in mosquito it is sporogony. So few dormant forms will that is hypnocytes, again they cause relapse, that is called as exoerythrocytic schizogony. So already saw in human cycle there are mainly two: pre-erythrocytic schizogony, erythrocytic schizogony, then gametogony, and in vivax and oval you may see exoerythrocytic schizogony, that is hypnozoites will again mature into the schizon form with milk. In mosquito it is gametogony. Sorry, in mosquito it is sporogony and the development of the gametocytes into gamete and fertilization, and then again the infective form sporozoite is formed. Now, what are the morphological forms seen in humans? So, in P. vivax, it is mainly ring form, it is thick and single. Then, tropozoid form, it is large, irregular, amoeboid. Then, third, scigon form, it is large, filling completely inside the RBCs and containing merozoites. Then, gametocyte, it is spherical, light blue cytoplasm. And then, RBCs are enlarged with the Schaffner's dots present in the RBC in P. vivax species. In P. falciparum, mainly it is ring form, which is thin or delicate, and many sometimes multiple rings can be seen inside the RBCs, and sometimes you can see headphone appearance, a cold forms. Then tropozoite is not seen, chrysont is not seen in P. falciparum. Then gametocyte, they are banana shaped or crescent shaped, and they are having pink cytoplasm. RBCs are usually normal and they contain Morris dots. So these are the pictures of P. vivax, ring form, tropozoite, schizont and gametocytes you can see in the PBS. Then these are the Plasmodium falciparum ring form and gametocyte form you can see inside the RBCs. And then in P. malariae, especially it is late tropozoite showing band form. Then P. oval, it is RBCs are oval and sometimes they are crenated. So pathogenesis. So there is mainly triad of fever, anemia and splenomegaly. So fever is because of the liberation of merozoites in the RBCs. Then anemia because of the destruction of erythrocytes, and splenomegaly because of the hemolysis, rapid hemolysis. The RBCs go to spleen and then spleen enlarge in size. So mainly triad of these three. Then fever mainly consists of first cold stage, patient will feel shivering and rigors, then hot stage, that is increased temperature, and then sweating stage. Complications of malaria is seen mainly in P. falciparum. They are also called as cerebral malaria or black water fever. In Vivax, it is sometimes Vivax and oval showing relapse because of the exoerythrocytic schizogony. Then P. malaria and knowlesi, they are mainly causing nephrotic syndrome, and chronic complications of malaria, it is tropical splenomegaly syndrome. So malignant malaria, what are the complications? So these are cerebral malaria. This we will see in detail. Then pernicious malaria, that is, it consists of black water fever, algid malaria, septicemic malaria, and the other systems are also involved. So RS is showing pulmonary edema or adult respiratory distress syndrome. Then sugar is low, so hypoglycemia. Then kidney is involved, that is renal failure, bleeding, diathesis. Then severe jaundice, anemia, and acidosis. So we'll see cerebral malaria. So it is due to plugging of brain capillaries by sequestrated parasitized RBCs leading to vascular occlusion and cerebral anosmia. So here in this picture you can see these RBCs are getting attached to the endothelium of the capillaries, and then because of that sequestration there is clot formation, and because of that there is hypoxia, decreased oxygen supply to the brain, and then it causes cerebral malaria. So patient will have symptoms like convulsion, and patient may go in coma, or in cerebral malaria there may be death of the patient. So cerebral malaria causes anemia, thrombocytopenia, then encephalopathy, retinal hemorrhages, hepatosplenomegaly, hypoglycemia, then metabolic acidosis, renal dysfunction, and sometimes 15 to 20% death. Then second, pernicious malaria. It is characterized by black water fever, algid malaria, and septicemic malaria. So black water fever means because of the sudden intravascular hemolysis after the fever, there is hemoglobinuria and the urine is dark in color. That is why it is called as black water fever. And it is mainly seen in P. falciparum. Algid malaria, there is cold clammy skin, then hypotension, peripheral circulatory failure, and shock symptoms. Septicemic malaria, high degree of prostration, high grade fever with dissemination of the parasite to various organs, causing multi-organ failure. So here you can see the urine color is dark, that is seen in hemoglobinuria. It is almost dark brown or black in color. Then patient is having hepatosplenomegaly, so liver and spleen are enlarged. Patient is having jaundice, it is mainly type of hemolytic jaundice, and then patient goes in renal failure. Now we will see laboratory diagnosis of the malaria. So these are divided into two, that is microscopic test and non-microscopic test. So in microscopic test, they are mainly peripheral blood smear, that is gold standard for the diagnosis of the malaria, that is thick smear and thin smear. Then fluorescent microscopy, quantitative Buffy coat examination. In non-microscopic test, it is antigen detection, and sometimes culture and molecular can be done. Peripheral blood smear. So it is simple and gold standard for confirmatory test for the malaria. So specimen is mainly either venous or capillary from the earlobe or from the finger, or in children or infants it is grade 2 or hill. Then time usually after the height, after few hours of the height of the fever and before taking anti-malarial drugs. And it should be at least twice till you don't see the parasites. So now types of peripheral blood smear, they are thin and thick smears. So in this picture you can see thick and thin smear. So first thin smear should be screened, and if you don't see parasites then you can go for thick film, or in some PSCs or some areas directly they go for the thick film because it is mainly sensitive. So for thick film, it is one large drop you have to take 2 to 3 drops in the center of the slide, and then you have to just make a circle with help of coin or with help of another slide, dry it and then stain it. Thin smear at the edge of the slide, you take 1 to 2 drops of blood, and with help of another spreader slide, you have to spread that drop forward so that you will get tongue shaped smear. So now staining, there are two methods, field stain and Leishman stain. So in field stain, you have to fix the smear after drying. So fix with methanol or with spray, then dry, then put that slide in a few dips into the field B, wash with water, then field A, few dips, and then wash with water, dry in air, and put oil and see under oil immersion lens. In Leishman stain, you have to flood the smear with Leishman stain. You don't have to fix the smear because the methanol is already present in Leishman. Then buffer added, and then wait for 5 minutes. Then wash with water, and then dry and see under oil immersion lens after putting oil. So first, thick smear. So advantages, it is helpful when the parasite count is very low, 5 to 10 parasites are there, still you can see thick smear. So in thick smear you just see the parasite forms, the RBCs are destructed, so you don't have to see, you don't see RBCs. In thick smear you can do quantification of parasites and demonstrate malarial pigments like Schaffner's dots or Morer's dots. The disadvantage here, species identification or morphological forms identification is not possible. In thin smear, advantages, it is useful in species identification. All the morphological forms you can demonstrate, or the RBC changes you can demonstrate in thin smear. But disadvantage, it is having low sensitivity, that is you need more than 200 parasites per microliter of the blood. So when the parasite count is very low, you cannot, mostly you don't see parasites in the thin smear. So this is picture showing thick film. So here you can see very few RBCs, but mainly parasites. You can see dots and nucleus. And the another picture, thin blood smear, you can see RBCs, then nucleus and ring, proper ring form. Whether it is thick, thin, you can see, or other morphological forms you can study in thin smear. So these are all Plasmodium vivax forms, ring form, tropozoite, schizont, and gametocytes. Then these are all morphological forms of P. falciparum. Mainly they are ring form and gametocyte, banana shape. Then in oval you see oval RBCs, and in P. malariae you can see band form of the tropozoite. These are all morphological forms of all the four species. Now, you can go for fluorescent microscopy. So in fluorescent microscopy, you have to just stain the smear with fluorescent dyes, that is acridine orange, and then you can see under fluorescent microscope. So there you can see greenish yellow fluorescence of the parasites. In quantitative Buffy coat, you have to take blood in capillary tube that is coated with the acridine orange fluorescent dye. Then in centrifuge that capillary tube, so it will have separation of Buffy coat. So Buffy coat is between junction between the cells, that is RBCs and plasma. So it contains mainly WBCs and lymphocytes. And then that you can see under UV light or under the oil immersion lens. So here you can see greenish yellow fluorescence of the parasitic forms. Now second test, nowadays it is performed many times. It is rapid antigen detection test. So here two antigens are used. So for all species, that is pan species, that is it is PLDH, then that is lactose dehydrogenase, and for Plasmodium falciparum it is mainly histidine rich protein 2, that is PFHRP2. So advantages, it is simple, but disadvantages you cannot differentiate other species in pan, and gametocyte stage or other stages are not developed, and it is not developed for the P. knowledge. So vector control for anti-adult, you have to use insecticides or you can use bed nets for anti-larval. Larvicide, use of mineral oil or Paris green to kill mosquito in stagnant water, or biological fish, that is Gambusia fish, can be used to kill the mosquito larva in a stagnant water. You can use that. Plasmodium species Plasmodium vivax, Falciparum ovale, Malaria knowlesi. Life cycle, mode of transmission, bite of mosquito, female Anopheles mosquito. Infecting agent, sporozoite. Human, pre-erythrocytic schizogony, merozoite zygont. Erythrocytic schizogony, merozoite zygont. Gametogony, male female gametocytes. Mosquito sporogony, microgamete plus macrogamete, form zygote, then oocyst, then kinate, and then the sporozoite. Pathogenicity, malaria. Febrile stage, cold stage, hot stage, sweating stage. Anemia, microcytic hypochromic because of the breaking down of RBCs during segmentation of parasites. Third, splenomegaly, enlargement of spleen. Complications of P. falciparum, black water fever, pernicious malaria, cerebral malaria, algae malaria, septicemic malaria, hypoglycemia, bleeding, renal failure, pulmonary edema, jaundice. In vivax, it is relapse. Lab diagnosis, specimen blood, PVS, thick smear, screening, thin smear, species identification. So morphological forms, ring form, tropozoite, zygont, gametocytes, four forms seen in P. vivax, and ring form and gametocyte forms seen in P. falciparum. Quantitative Buffy coat examination, rapid malaria antigen test.
Hope you find this session useful for your understanding and for your exams. Thank you.